Welcome. In this video, I will be discussing the cell wall of bacteria and archaea. In my previous video, I explained the plasma membrane of bacteria, which protects the cytoplasmic contents from the external environment. However, the cytoplasm of bacterial cells contains a high concentration of dissolved solutes, leading to a significant osmotic pressure, approximately 2 atmospheric pressure in a typical bacterial cell. To withstand this pressure and prevent cell lysis, bacteria need a second layer of protection, the cell wall. Additionally, the characteristic shape of the bacteria is due to the cell wall. All bacterial cells contains a cell wall except for mycoplasma. The bacterial cell wall is a rigid protective layer that surrounds the cell membrane. It provides structural support and shape to the cell, prevents it from lysis, and acts as a barrier against environmental stresses and harmful substances. So we can say that the cell membrane is like the tube and the cell wall is like the tire. The cell wall of bacteria is composed of peptidoglycan. The peptidoglycan, also known as murin, is a large mesh-like polymer composed of many identical subunits. The basic subunit consists of 3 parts: N-acetylglucosamine or NAG, N-acetylmuramic acid or NAM, that is connected by beta-1,4 glycosidic linkages. The short peptide chain of 4 alternative D and L amino acids are attached to the carboxyl group of N-acetylmuramic acid. The 4 amino acids are L-alanine, D-alanine, D-glutamic acid, and either lysine or the structurally similar amino acid analog diaminopimelic acid, or in short DAP. These basic units are attached side by side to form a very fine thread-like structure like this. They are arranged side by side or one above other to form a mesh-like structure like this. The backbones are then connected by a peptide cross bridges. The formation of peptide cross bridges, also known as transpeptidation, is crucial for the cell rigidity. Without this cross-linking, the chain would be unable to withstand the cell pressure. When Gram staining is performed, the bacteria exhibit 2 different types of Gram reactions. One type of bacteria takes up the primary stain, that is crystal violet, and appears purple under the microscope, while the other takes up safranin, a counter stain, and appear pink or red color. Based on this Gram reaction, bacteria can be broadly classified into 2 types, Gram-positive and Gram-negative. The cell wall of Gram-positive bacteria is generally thick, about 80 nanometer, and contains around 30 layers of peptidoglycan stacked on top of each other. Because of its thickness, the complex formed by crystal violet and iodine is not washed out from the cell by a brief alcohol wash. The cell appears purple or blue under the microscope after the Gram staining is performed. Peptidoglycan accounts for 40 to 80% of the dry weight of the cell. In Gram-positive bacteria, cross-linkage may occur through a short peptide interbridge, with the types and numbers of the amino acid in the interbridge varying from species to species. Additionally, the cell wall of Gram-positive bacteria contain teichoic acid, which consists primarily of an alcohol and phosphate. There are 2 classes of teichoic acids. One is lipoteichoic acid, which span the peptidoglycan layer and it is linked to the plasma membrane. And the second one is wall-teichoic acid, which is linked to the peptidoglycan layer only. Because of their negative charge, teichoic acid also function to bind calcium and magnesium for eventual transport inside the cell. On the other hand, the cell wall of Gram-negative bacteria is more complex than that of Gram-positive bacteria. It is generally thinner than the Gram-positive bacteria, about 2 nanometer. That's why when alcohol is used, the crystal violet and iodine complex is washed out and the bacteria become clear. To increase the contrast, a second dye called safranin is used, and as a result, Gram-negative bacteria look pink or red under the bright field microscope. Peptidoglycan may constitute 5 to 10% of the cell's dry weight here. In Gram-negative bacteria, peptidoglycan cross-linkages occurs through the peptide bond formation between the amino group of DAP and one glycan chain of the carboxyl group of the terminal D-alanine on the adjacent glycan chain. The peptidoglycan layer is not as thick as that of Gram-positive bacteria, which is why a third layer known as the outer membrane is found in Gram-negative bacteria but is absent in Gram-positive bacteria. The outer membrane plays a crucial role in reinforcing the structure of the gram-negative bacterial cell wall. In gram-negative bacteria, the outer membrane makes up a significant portion of the cell wall. It's just like a second lipid bilayer, but it is distinct from the cell membrane because it is not composed solely of phospholipid and proteins. Instead, the outer membrane also contains polysaccharide, which are linked with the lipids to form a complex known as the polysaccharide layer, or LPS for short. The outer membrane consists of LPS, lipoprotein, and phospholipids. The inner half of the outer membrane is composed of phospholipids and lipoproteins. Lipoprotein function as an anchor, tying the outer membrane to the peptidoglycan. LPS replaces much of the phospholipids in the outer half of the outer membrane bilayer, as you can see in this picture. LPS composed of three components: lipid A, the core polysaccharide, and O-polysaccharides. The O-polysaccharides typically contain galactose, glucose, rhamnose, as well as one or more dideoxyhexose. These sugars are connected in 4 or 5 member sequences, which are often branched. When the sequence repeat, a long O-polysaccharide is formed, which is generally recognized by the host antibodies. The core polysaccharide consists of 2-keto-3-deoxy-octonate and various 7-carbon sugars, glucose, galactose, N-acetylglucosamine, and it is connected to the O-polysaccharides. The lipid portion of LPS, called lipid A, is not a typical glycerol lipid. Instead, the fatty acids are connected through amine groups from a disaccharide composed of glucosamine phosphate. The disaccharide is attached to the core polysaccharides through KDO. Fatty acids commonly found in lipid A are listed here. Some gram-negative bacteria, including species of Salmonella, Shigella, Escherichia, are pathogenic to humans and other animals due to the toxic outer membrane components. The toxicity is mainly associated with the LPS layer, particularly the lipid A. It is released into the bloodstream when the bacteria die, especially due to the broad-spectrum antibiotic treatment. The term endotoxin refers to the toxin component of LPS. Some endotoxins cause severe symptoms in humans, including gas, diarrhea, and vomiting. The gap between the cell membrane and the cell wall is called the periplasmic space. This gap is most easily observed under electron microscope in gram-negative bacteria, but it is rarely observed in gram-positive bacteria. And this space contains the cell wall of peptidoglycan, the many digestive enzymes, many transport proteins. Peptidoglycan is a key biomarker for bacteria, but it is absent from the cell wall of Archaea. Unlike gram-negative cell wall, Archaea don't have any outer membrane. Instead, a variety of chemistries are found in the cell wall of Archaea, including polysaccharides, proteins, and glycoproteins. The cell wall of certain methanogenic Archaea contains a molecule that is remarkably similar to peptidoglycan. A polysaccharide is called pseudomurin. The backbone of pseudomurin is composed of alternating repeats on N-acetylglucosamine and N-acetylmuramic acid. The glycosidic bonds between these sugar derivatives are beta-1,3, and the amino acids are all in L-stereoisomer. The cell wall of Archaea have a thick polysaccharide wall composed of polymers of glucose, glucuronic acid, galactosamine, uronic acid, and acetate. Extremely halophilic Archaea, known as Halococcus, contain sulfate in their cell walls. The negative charge of the sulfate binds to the salt concentration of sodium present in the habitats of Halococcus, such as the salt evaporation ponds and saline sea and lakes, helping to stabilize the cell wall in such strongly polar environments. Some bacteria like mycoplasmas, which cause several infectious disease in humans and other animals, and the thermoplasma group of Archaea naturally lack the cell wall. These bacteria can survive without cell walls because they either have usually tough cytoplasmic membrane in case of Archaea or live in a osmotically protected habitats in case of mycoplasma. Most mycoplasma have sterile in their cytoplasmic membrane, which likely functions to add strength and rigidity to the membrane. Similarly, how sterol function in the cytoplasmic membranes of eukaryotic cell. Finally, I will discuss the medical significances of the bacterial cell wall. The bacterial cell wall is an excellent drug target. Several antibiotics that target the cell wall biosynthesis are available for treating bacterial infection. In fact, the first antibiotic penicillin discovered by Alexander Fleming also targeted the cell wall biosynthesis. Penicillin contains a beta-lactam ring, And the other antibiotic that contains these ring are also called beta-lactam group of antibiotics. The beta-lactam antibiotics are inhibitor of cell wall biosynthesis. An important feature of bacterial cell wall biosynthesis is transpeptidation, the reaction that results the cross-linking between the two glycan chain. The transpeptidase enzyme bind to the penicillin or other beta-lactam antibiotics so that the transpeptidase are called penicillin-binding proteins. When penicillin-binding protein binds with penicillin, they can't catalyze this transpeptidation reaction, but the cell wall synthesis will continue. And as a result, the newly synthesized bacterial cell wall is no longer cross-linked and cannot maintain its strength. Additionally, the antibiotic PBP complex stimulates the release of another enzyme, autolysins, the enzymes that digest the existing cell wall, leading to a weak, self-degrading cell wall. Eventually, the osmotic pressure difference between inside and outside of the cell causes lysis or cell death. In contrast, vancomycin, another cell wall synthesis inhibitor, does not bind to PBP, but binds directly to the terminal D-alanine peptide on the peptidoglycan precursors and effectively block the transpeptidation. Because the cell wall and its synthesis mechanism are unique to bacteria, beta-lactam antibiotics are highly selective and are not toxic to the host cells. However, some individuals develop allergies to beta-lactam antibiotics after repeated exposures. Cephalosporins are another group of clinically important beta-lactam antibiotics. Cephalosporin produced by fungus Cephalosporium species differ structurally from penicillin. They retain beta-lactam ring but have a 6-member ring instead of 5-member ring. Cephalosporins have the same mode of action as penicillin. They bind irreversibly to penicillin-binding proteins and prevent the cross-linking of peptidoglycan. Clinically important cephalosporin are semisynthetic antibiotics with a broader spectrum of activity than penicillin. And most important of all, cephalosporins are typically more resistant to enzymes that destroy beta-lactam rings, known as β-lactamases. For example, ceftriaxone is highly resistant to beta-lactamase and has replaced penicillin for the treatment of Neisseria gonorrhoeae, because many strains of Neisseria gonorrhoeae are now resistant to penicillin. And last, I will discuss about one enzyme called Lysozyme present in our body secretion like saliva, tears, and break this bond, and as a result, bacteria will die. These enzymes actually protect our body from those bacteria. So that's all for now. If you found this video helpful, please like, share, and subscribe to my channel for more content. And finally, thanks for watching.